Hello everyone, welcome back to our channel. Today we will discuss about the gram-positive and gram-negative bacteria. In the previous class, we have understood that the basic morphology of bacteria, and today we are understanding what is gram-positive and gram-negative bacteria. Secondly, we will understand the difference between the gram-positive and gram-negative bacteria. And thirdly, we will understand 5 names of gram-positive bacteria and 5 names of gram-negative bacteria. Let's start the class. See here, firstly, gram-positive and gram-negative bacteria. The differentiation are coming by the gram staining. It's a one type of differentiate staining which help to giving us the gram-positive and gram-negative bacteria. These strains are developed firstly 1884, hence Christian Gram. So that this name is Gram stain, or we can say that Gram staining. In this video, we are not going to discuss about the Gram staining. We are discussing about the bacterial differentiation. See, if you are noticing here, so that the board is completely blank. If you are noticing here, firstly that is the gram positive and that is gram negative. Here I am just taking the column so that whenever you are understanding this picture, you can easily differentiate the gram positive and gram negative bacteria. And in the way I am teaching, you will never forget that. See, here is the column that is gram positive, and second one should be gram negative. So that firstly, see here, gram positive is thick, and gram negative is the thin. See here, that is the thin, in nature, and that is thin. Gram positive bacteria, peptidoglycan. If you are noticing that here, peptidoglycan is too much thick. Here is the peptidoglycan. See here, too much thin. Peptidoglycan, thick. Peptidoglycan, here is a thin. That is the second differentiation. Thirdly, if you are noticing here, here is a teichoic acid present in gram positive. Here does not contain any kinds of teichoic acid. So that teichoic acid, teichoic acid here is a present, here is absent. After that, if you are noticing here, here is a LPS, means lipopolysaccharide, where the polysaccharide are attached with the lipid, that is called lipopolysaccharide. It basically help to creating this membrane. Basically, gram positive and gram negative bacteria differentiation are coming to the cell membrane differentiation. They both are the cell membrane. That is the cell membrane of gram positive, and that is the cell membrane of gram negative. Okay, so that there is LPS, lipopolysaccharide, in the gram positive, that is absence, right? And into the gram negative, here is the LPS is present, so that here is present. After that, if you are noticing here, here is a periplasmic space is present, and here does not contain any kinds of periplasmic space. So that, see here, periplasmic space here does not contain, gram positive bacteria does not contain any kinds of periplasmic space, and here is the present. After that, if you are noticing here, there is a cytoplasmic membrane that is present in both. See here, so that is not a differentiation. Whenever you will do the gram staining, then you will find out one thing, that the gram positive bacteria is blue or violet in color, and the gram negative bacteria, that is a pink or red in color. Okay, after that, if you want to differentiate it, just write down here some example and here some example. If I am taking some example of gram positive bacteria, that should be Streptococcus aureus, the Staphylococcus aureus, Streptococcus pneumoniae. Okay, and if I am taking the example of gram negative, that is E. coli and Klebsiella, they all are the Enterobacteriaceae, and they all are coming under the gram negative bacteria. So that is really very much easy thing. We can easily understand the differentiation of gram positive and gram negative bacteria. That is very important question for your board exam, for your interviews, as well as for your a various type of competitive exam also, that is very much important question, gram positive and gram negative bacteria. There might be various type of MCQ are present within the gram positive and gram negative bacteria. Okay, after that, if I am coming noticing here, that is gram positive bacteria. Firstly, Staphylococcus aureus. Staphylo, defining that, we have understood into the first lecture. Staphylo means there should be grapes, right? Grapes like structure. And coccus, defining that should be round. So that Staphylococcus should be looking like that. Okay, after that, streptococcus. What do you mean by strepto? We have understood that strepto defining the chain, right? Strepto defining the chain, and coccus defining the round shape. So that streptococcus pneumoniae, that is also round in shape and chain in arrangement. After that, staphylococcus poison. After that, cornybacterium diphtheria, that basically causes the diphtheria disease. Cornybacterium diphtheria, it is looking like a Chinese letter shape. We have understood that is looking like a Chinese letter shape. After that, see here, that is a bacillus anthrax, that basically causes the anthrax disease. Okay, that's all are the gram positive bacteria. After that, if you are noticing here, that is the gram negative bacteria. It's also important whenever you will attend any kinds of viva or any kinds of interview, there might be accessible question is write down or say something gram positive and some gram negative bacteria, so that you have to say it. There is nothing option. See, if I am coming that gram negative bacteria, most of the pathogenic bacteria are under gram negative bacteria. Okay, see here, firstly, E. coli or Escherichia coli, it is most prominent in nature. How it look like? E. coli is the bacilli and gram negative bacilli, so that we are saying that GNB. Okay, so that whenever you are doing the work in various type of laboratory, they will uses these kinds of small name GNB. After that, see here, GPC, that is gram positive cocky. Okay, that's all are the short form. Okay, see, after that, Klebsiella. E. coli and Klebsiella are most prominent bacteria into various microbiological laboratory. These two bacteria are looking like same. Their morphology is also same. They all are GNB. It is also GNB, gram negative bacilli. But there is only one difference. These questions are also important for your viva interview purpose. Okay, there is only one difference. E. coli are motile, means E. coli are flagellated, and Klebsiella are not motile in M, not motile. Okay, so that is only one difference that is important. You need to understand that. Okay, after that, if I am coming to the another gram negative bacteria, that is Vibrio coli, that basically causes the cholera disease. See, Vibrio cholerae is looking like coma shape. We have understood that. So that arrangement of bacteria we have already understood, that is cocky, that is bacilli, that is vibrio, that is Chinese letter shape. Here you are seeing the application of these kinds of whole bacteriology as well as these kinds of whole arrangement of bacteria. Okay, after that, Trachinema pallidum, that basically causes the syphilis, sexually transmitted disease, syphilis. Okay, after the Nigeria meningitis and GTS. Another one, Nigeria basically there is a two pathogenic species. One is Nigeria meningitis and another one is a Nigeria gonorrhea. Nigeria gonorrhea causes the gonorrhea disease, and meningitis basically causes the meningitis. Okay, but Nigeria meningitis is also under the gram negative bacilli, gram negative bacteria. Okay, if you are noticing here, here is the gram positive all bacteria and here is the gram negative all bacteria. There have also lots of bacteria regarding gram positive and regarding gram negative, but I am saying you 5 example. If you are remembering the five example, that is enough for you all guys. Whenever you are ensuring the systemic bacteriology, then you will understand that into the morphological features, there you will find that first morphology you have to say it is gram positive or gram negative. Second morphology you have to say is it cocky, is it bacilli.
Third morphology, you have to say, is it motile or immotile? Fourth morphology, you have to say, is it sporing or non-sporing? Fifth morphology, you have to say that it is capsulated or non-capsulated. And whenever I will take the class of systemic bacteriology, then you will understand it. Okay, if you want to join into our batches, they have the free batches. Just check the description box, you will get the whole kinds of information, and share the video and subscribe our channel. It will help you a lot, and it will help you to enhance your knowledge in the field of laboratory technology. Thanks a lot for listening as well as watching.